So I'm a research assistant working under Kati Bonner, and that I got an immense opportunity to introduce Professor Shankar Subramaniam. He's working as an professor of bioengineering, computer science and engineering, cellular and molecular medicine, along with it nanoengineering at University of California, San Diego. His research interests are in systems biology and medicine. I hope you'll enjoy his session.
Thank you very much. I'm assuming. Great. So Kaki Borner asked me to speak today. I've had a lot of interest in HubMap, even though I'm not funded by HubMap to do things, but I have tremendous amount of interest in HubMap. So I thought I'll talk about one of my favorite things with HubMap, which I've been plaguing HubMap folks about, which is the following. When you talk about tissue mapping and cell mapping to a given anatomical organ or a region in human physiology, we often ignore completely the aspect of development and the aspect of integrative functional units. And so this has really been of great interest to us in terms of various human physiological tissues and organs. The question is the following. When we had just heard about the kidney, we heard about other organs. I mean, clearly, during the developmental process of a child going from a childhood to an adult, there's an evolution of the organ in some meaningful ways, and this is not captured in HubMap. It's very difficult to capture it because we are really talking about trying to look at the progression or the development of a given tissue and organ in human physiology. And this is an extremely complex issue. So what I'm going to tell you today is a perspective on skeletal muscle. We have done a lot of work on skeletal muscle in terms of looking at, I'll look at it from an anatomical, embryological perspective to tell you what happens in the skeletal muscle growth. Then I'll explain this to you from a point of view of a cellular perspective. How does a muscle cell evolve from a pluripotent stem cell? And then I'll give you a molecular perspective where I want to tell you the key factors and the key functional processes that are changing as a function of development of the muscle. Why do we need this? We need this because of 3 or 4 reasons. The first reason is we want to look at development and developmental disorders in human physiology, and we want to look at how the development is altered in the process due to various defects or various either genetic or other environmental defects. I mean, you've seen several examples, for example, in brain development, there's autism. You look at several examples of tissue development, heart development, where there are defects in the heart, function. And so there are, many, many things that can happen during development. So we need to have a developmental perspective in terms of describing a tissue or an organ in a HubMap context. The second reason why we need to look at development in a meaningful way is with the advent of new technologies, including stem cell-based technologies and using CRISPR-based perturbation methods, we are in a position to be able to redress any issues that arise in development due to genetic or epigenetic disorders. And so this provides us with an opportunity to understand the contextual understanding of where a molecule fits in the developmental axis of a tissue or an organ. So this is what I want to go over today at some level of detail with you. And the question I want to pose is what makes a muscle? I want to go through the developmental axis. I want to go beyond a HubMap perspective, which is a kind of a steady state perspective of a canonical organ that is in, few human beings that are averaged over from imaging modalities and other modalities. So I'll try to give you a multi-scale view of this. The first thing I want to give you is a cartoon figure. And today, in fact, with stem cell technology being there, it is a stem cell parental advice. You can be anything that you want to be when you grow up, but being a muscle is good. And so this is really, I want to walk you through, this is more a fun thing to tell you. It's past 11 p.m. in California where I am. So I thought I'll just tell you something to keep it activated. Let me go to an embryological perspective in terms of development. I'm sure some of you may know this to some extent, but many of you may not know this. This is actually taken from a mouse embryo. if you look at day 10.5, a mouse embryo, which carries the MYF5 lineage trace. So MYF5 is a transcription factor, which is coded in red. And if you look at A, you can see that there is an increase in MYF5 on day 10.5. And this expression can be observed in a region called the presomatic mesoderm, the somites, and several head structures. Now I'm going to spread this, elongate this guy along this C, along this axis C. In B, I'm just mimicking this, showing you that in different regions, the caudal regions, the rostral regions, there is a differential effect of different factors like Wnt and FGF. And these are really growth factors that are really instrumental in defining the germ layer. But I'm going to show you more in detail along the axis. If I kind of look through a cross section of the embryo, then if you look at the caudal region, which is where the early somite exists, you have activation of the Wnt signaling pathway and the introduction of BMP4, which essentially gives rise to the combination of morphogens changing and giving rise to morphogenesis. In fact, morphogens from various domains in the embryo specify the early somite form of what's called the sclerotome, which is given by this thing called SC. And the dermomyotome is the one which is here, and that's what gives rise to the specifying the muscle. And when Wnt is secreted from the dorsal neural tube and the surface ectoderm along the morphogen gradient with BMP, and then that is when it signals or initiates the formation of the specific mesoderm. And this one gives rise to the ventrolateral lips, which are these regions, is where the first mature myotome arises from that point. And this is dependent on PAX3, which is a transcription factor. And then from there, we give rise to limb muscles. And this is well studied in mice embryo. In fact, people have done very good imaging studies on the mice embryo, not on human. We don't have the human perspective, but the understanding is that mice and human have a lot of similarities. So if you look at the perspective from going from embryogenic, early embryonic system all the way down to a skeletal muscle, which you know is organized in the form of fibers, you go through various stages and various transcription factors. In fact, the embryonic progenitors, the earliest activators are this PAX3, which is the transcription factor I told you in the dermomyotome that originates. And then you get the satellite stem cells. And in this case, you activate MYF5, I pointed to MYF5, and then you get MyoD. And then when you go towards the committed satellite stage, you start expressing PAX7. And I'm going to show you all of this from a very different perspective in a few minutes. And then when you go into myoblast, you start getting increased MYF5 expression, increased MyoD expression, which persists all the way into myocytes. And when you go into mature myocytes and myotubes, you start getting MyoG, which is a transcription factor, and MRF4, which is also a transcription factor. And these define the skeletal muscle. So going back to Rahul Sathija's talk about single cell markers, if I'm doing a single cell experiment in a muscle development, I would look for these markers as being the key markers that discriminate between the satellite stem cell case, the committed satellite case, the myoblast stage, the myocyte stage, and the myotube stage. So this is really the kind of commitment that happens in human physiology and in mammalian physiology in general. So here's the perspective of the same thing I showed you. This is a schematic of the skeletal muscle in the satellite niche. When you get into the dermomyotome, I think I pointed out to you the VLLs, and the VLLs show structures like this, which really form when you expand on this. There is the myonucleus, there is the basal lamina, there's a satellite cell, which is a precursor for the muscle cell myocytes, and then there's a muscle fiber, and then the blood vessel comes through here, and the motor neuron activates the cell. And I'm going to walk you through this later in my presentation in just a little while. So if I look at the satellite stem cell, which I just showed you, has symmetric division and asymmetric division. When we do the symmetric division, it goes into the myonuclei. asymmetric division takes into basal lamina and sarcolemma, and this gives rise to the satellite myogenic cell. The differentiation, or in fact, it's very, very interesting because when you look at normal muscle, you have a tear in your muscle, for example. This tear in your muscle is repaired. How is it repaired? I mean, this is exactly what happens. In fact, your muscle cells can differentiate and then can return to quiescence, and then once the muscle is grown, and the quiescence markers for the skeletal muscle or transcription factors like PAX7, MYF5, MyoD, MyoG. When you have a PAX7 positive, MYF5 minus, MyoD minus, MyoG minus, the tissue tells the cells, stop differentiating. You already formed a mature muscle. And so that is really where the quiescence is achieved. For proliferation, you need to have activation of various markers at various stages. And for differentiation, you again have various markers at various stages. So today, if I had a dynamic ability to monitor how my muscle differentiates into, how my stem cells differentiate into a muscle, I'll be able to monitor it using these markers, scope of these markers, and then look at what happens. And this is exactly what I'm going to do in just a minute with you. So what we did was we said, let's do the following. Let's take stem cells, induced pluripotent stem cells from human subjects. We take it from different human subjects. And then we are trying to now differentiate them into myocytes using the same protocols that is used in vivo in a mammalian system. So we really want to, try to make a muscle, try to lineate, specify, or differentiate into a muscle using exactly the same logic that is used in vivo situation. So we took three different human subjects. In fact, this is so exciting, and this really has bearing on HubMap because it tells you that not all human beings produce muscle at the same rate. Not all human iPSC cells produce muscle or mature muscle in the same way. They require distinct factors. I'm going to show you that. Let's take these three different human iPSC cells. One is TL, the other is SCB, and the third one is LEP. I'm going to just for the sake of convenience call them LEP, SCB, and TL. And this is actually, I'm showing you, and I don't know if you can see it well, it's a smaller image than I wanted it to be. You can walk yourself through day 3, day 5, day 8, day 12, day 16, day 19, day 25, day 30. We monitored all these days, and we monitored various markers. We monitored markers like, for example, we monitored MYF5, MyoD, MyoG, and Desmin, which is a genetic marker. And you can see clearly not all stem cells behave exactly in the same way. Some stem cells are better at producing muscle. For example, LEP in day 30 is completely, it differentiates into muscle, whereas TL and SCB do not differentiate fully into muscle. And we asked the question, what are these factors which make this development so unique and interesting in terms of specifying the muscle? And this is what I'm going to walk you through for the next 10 minutes. I'm going to show you what factors make one stem cell different from another stem cell, and what are the implications for human diseases? Because this is really the key aspect. For example, imagine somebody has muscular dystrophy of some kind or has some other disorder, Lou Gehrig syndrome, for example. And then how do we figure out what are the factors that are involved? So this is what I'm going to walk you through. In fact, we did a very interesting analysis of these things. We took each of these cells, and at each of these days, we did transcriptomic measurements using RNA sequencing. And we tried to ask the question, what are the molecular players who are responsible for various events across these different time points in the TL cell, in the CD cell, and LEP cell. So we use next-gen sequencing, RNA sequencing, to monitor go across this time. So when I do this, in fact, this is one of my graduate students, Priya, who's also an artist. So she decided to draw a beautiful painting on which she wanted to implant all these markers. If you take each of these cells, T, S, and L, remember, let me go back one. You remember this is TL, I call this S, and I call this L. And based on just looking at the imaging, you can see clearly L produces the largest number of myocytes, aligned fibers, which are marked in red by using PAC7, which I told you previously exists throughout the mature myocyte. And so we can see clearly L is more better than S is better than T in terms of specifying myogenic lineage. So what we did, Priya and others did, was to ask the question, let's longitudinally monitor what happens across time in terms of creation of a muscle. So if you look at the stem cell, the primitive stem cell has, embryonic markers, OCT4, which is POW5F1, NANOG, SOX2, KLF4, and as you know, these are all stem maintenance transcription factors. In order for a cell to go into a lineage, specific lineage, and go away from a stem cell, it's got to reduce or suppress these factors like OCT4 and NANOG specifically. You can see amongst the factors T, S, and L, each of these lines here, each of these horizontal bars here represents one cell across time points. Each vertical point is a time point. Horizontally, it's each type of cell. You can see already L is the one that suppresses OCT4 and NANOG much more than T and S, and therefore it's more poised to become a muscle cell. And if you continue this thing, in fact, the precursor, I don't know how many of you know stem cell, basic stem cell biology. You go through in human physiology, when you go from embryogenesis towards lineage specification, you go into various germ layers. You go into mesendoderm, endoderm, and ectoderm. In fact, all of these muscle precursors go through the mesoendoderm. And you can see that the mesoendoderm specification is done through transcription factors like EOMs and T and MSG-N1. And again, all these three different cells are subject to going towards a mesoendoderm like fate. However, when you go beyond this point towards the end stages, the first cell, which is the first two cells, which is the T cell and the S cell, they are going more towards an endoderm-like lineage. And then they also go towards a more cardiogenic mesoderm lineage. And whereas only L, which is predominantly going towards a paraxial mesoderm lineage, which I showed you in the embryo, the paraxial mesoderm is the one that specifies the muscle. And so this is predominantly the path that a muscle takes, a myogenic pathway takes. And so we can see that this stem cell is the one that goes into this fate. You can ask me, why do I care? I care because in different humans, we now have the ability to take either skin cells, which is fibroblasts, or blood, transform them into induced pluripotent stem cells. And then we can actually monitor this process of specifying a muscle outside the human physiology. And then this can help us really look at repair mechanisms that we can induce either pharmacologically or genetically. So let me go on to show you what happens. So if you look at these different distinct mechanisms that go towards this specific myogenic lineage, in the very early stages, I'm looking at T, S, and L. Now I'm looking at them in terms of a column. If you look at factors that specify, for example, what happens in a muscle specification, the FGF pathway has to be increased or enhanced. The Notch pathway has to be enhanced. The Hedgehog pathway has to be enhanced. And the TGF family beta pathways have to be suppressed. That's what defines a muscle in embryogenesis. So when we look at this, in the early stages, you can already see that only L is the one that is the dominant one that goes towards the muscle lineage, lesser extent S, and even lesser extent T. And the mid lineage in neuronal, mainly we go into specification of a muscle. Now, what specifies a muscle? Dystrophin. Dystrophin is one of the largest proteins, and I'll come back to it and tell you a little bit more about it later. And dystrophin is increased in expression in the L lineage. And all the neuronal factors, calcium channel factors, which are also involved in the specification of the myogenic lineage, or it has more in the L than in the T or S case. And similarly, in the final late stage, sarcomere formation is really the key for specifying a muscle. And you can see factors like MY, myosin heavy chains, myosin light chain, troponin, myosin heavy chain. These are all increased in the L lineage, which tells us that we are going towards a mature muscle. How about neuromuscular junction factors, ACH receptor? Again, increased in the L lineage across. So what I've shown you so far, is that starting from distinct stem cell lineages, we'll be able to define a muscle. And we not only know how to define a muscle, we know what factors can be tuned to define a muscle. In the paper that I've referenced in this, we showed that in order to make, for example, a T lineage go into a muscle, we can actually induce transcription factors that are activated in the L cells, and we show that it immediately starts making muscle. So this gives you really not only a dynamical perspective on the mechanism which is transforming a stem cell into a muscle. Let me go to the next slide. I can then look at also the functional pathways that are involved in each of these cases. For example, we used a procedure called a short-term expression miner, and then we can show different pathways that are involved. For example, if you take this particular all genes that are going up linearly, and you can see that in the L case, all the genes are increased or involved in specific muscle specification and metabolism. And so we really can start piecing out machinery involving various pathways that are consistent with the progression towards a muscle. Now, let me now walk you through from here. Once I've specified a muscle, the question that automatically comes about is the extension of this functional idea to look into what does the muscle do. In fact, HubMap, one of the key questions that HubMap tries to pose is that when you look at a kidney, it tries to define functional tissue units in the kidney, which are responsible for specific function. So we can ask the same question of the muscle. Can we define the larger scale functions that a muscle does? And can you define the molecular factors that are involved in the muscle specification so that we can go on to look at whether these molecular factors are activated in a canonical skeletal muscle or in an injured muscle or a repairing muscle? So this is a question that we try to ask. So what does a muscle do? A muscle does just 4 things. That's it. What are the 4 things that a muscle does? It essentially collects the signal from the neuron in the neuromuscular junction. This is what I told you about the ACH receptor a minute ago. And this is called excitation activation. Muscle senses electrical signal and induces excitation activation, which involves a number of ion channels and transporters. And this is communicating immediately after it gets as excitation activation. It triggers the signal for mechanical activation of a muscle and force transduction. And this is all done through intermediary machinery like calcium containing machinery, phosphates, ATP, which is involved in metabolism, and protons. Then the next family is the mechanical family, and I'll walk you through mechanical family because it's really, really exciting to understand how a muscle works. And then there's a metabolic family, which provides all the energy required. In fact, I'm sure you have seen this. If you look at a sprinter or a long distance runner, they tell them, oh, you've got to have this type of muscle or that type of muscle. You need to provide energy. You need to have a glucose drink, so that you can immediately get energy out of it. So that's what the metabolic family does. It provides energy for mechanical family. Signal transduction provides the ATP, and this ATP is used for various functions like force transduction. And then there's a signaling and translation family. And many of you may have heard about remodeling of the muscle fiber. In fact, it's a dynamic remodeling of the muscle fiber. When you go from a glycolytic machinery to an oxidative machinery, you go from a fast fiber form to slow fiber form. And then this is done through the signaling and translation family, which essentially signals to the nucleus, to the chromatin, to express specific type of signals which will make it into a slow fiber or a fast fiber, etc. Now I'll walk you through this in the next 10 minutes so you get a perspective of this. We did an extensive analysis of of each of these 4 families. And in fact, we have a review. There's a 50-page review that is published in Interdisciplinary Reviews in Systems Biology and Medicine, which provides really a comprehensive view of each of these functional families. And if you look at excitation activation family, this is the neuromuscular junction. And the neuromuscular junction essentially provides the electrical impulse. This electrical impulse functions through various channels and receptors. There are channels, calcium channels. There are Shankar pumps. There are ryanodine receptors. And these really change the electrical activity. Muscle, as you know, is one of the excitable cells. And so it changes electrical activity, changes the mechanical activity. And it tells the muscle cell, that there's a signal coming from the neuron, from the brain. And so this is how you really move your hand. When I move my hand, when you do things mechanically, this is exactly what happens. In fact, the schematic representation I have here shows excitation contraction coupling of the triad junction, which is DHPR, RYR1, and Shankar pump. And these act in concert together to change calcium signaling and current, and this calcium current alters the storage and other mechanisms, which essentially initiates the activation of the muscle. Calcium is the key to signal the activation of the muscle. How do we get activated? The very first thing that happens with calcium activation from excitation activation is the calcium binds to troponin C, the troponin family, which then activates tropomodulin. Tropomodulin activates nebulin and F-actin and CAB-C, and these are all the actin subfamily proteins. These actin subfamily proteins act in concert with the myosin subfamily proteins, which is myosin heavy chain, myosin light chain, titans, myomasins, and other C proteins. And these in turn activate the Z-disc family in the muscle. I didn't have time to tell you about the anatomy of the muscle. It activates the Z-disc family, which involves desmin, plectin, actinin, etc. And this in turn communicates with the transmembrane family. This already communicates to the membrane proteins, which are dystrophin, laminin, caveolin, F-actin, integrins. And then it tells the cell, hey, you've got to do something. You've got to morph. You've got to move. You've got to align. You've got to do contraction, excitation. And this in turn relates to the extracellular matrix subfamily, which is fibrinogen, collagen, fibronectin, versican, and this transduces into a force. And so this is really how the orchestration of force transduction happens in a skeletal muscle. The second function, in fact, I want to show this to you in a little bit more detail. I told you how is force transduced here. Here I'm going to show you how force is transduced. In fact, you've got the actin, tropomyosin, troponin, actin family of things. And then many of you have seen this before. This tropomyosin really acts through this complex network of proteins to move the muscle back and forth, to contract and expand and so forth. And in fact, when I go to the extracellular matrix, which I told you the extracellular matrix is communicating. The extracellular matrix has a beautiful set of proteins, which are large proteins, things which are involved like collagens and versican and hyaluronan. And these act in concert towards producing the excitation contraction action. And in fact, these extracellular matrix proteins and their localization is crucial in terms of muscle function. When you have a tear, muscle tear, in fact, there's a lot of times when you have this damage to the ECM matrix. Okay, now what happens to the cytoskeleton? The cytoskeleton is really involved with the membrane cytoskeleton. It involves this set of proteins called, dystrophins, dystrobrevins, and dystrophins. Dystrophins, in fact, many of you may recognize this dystrophin is one of the largest proteins, and it is very actively involved in force transduction. How many of you have heard about muscular dystrophy, Duchenne muscular dystrophy, or other types of muscular dystrophy? There, one particular subunit of this dystrophin is missing in those human subjects, in these human patients, because of genetic changes. As a consequence of the missing domain, your dystrophin doesn't function, which is what gives rise to muscular dystrophy in patients. And often, children die by the time they are 20. So in fact, it will be fantastic if we can find new mechanisms and models towards doing ways of replacing skeletal muscle with the proper dystrophin. And this is, in fact, there's a clinical trial underway to do exactly this kind of thing. Let me talk about the metabolic family for the next 5 minutes. And metabolism is the key towards muscle function. In fact, many of you know that, I want to tell you about two facets of the thing. A muscle metabolizes, in fact, muscle is instrumental in metabolizing glucose, sugars, and other things. And in fact, this glucose glycolytic machinery is what gives rise to instant energy in a skeletal muscle. You get, immediate energy in the muscle. For example, imagine you're doing running, you snatch a bottle of Gatorade or some electrolyte and sugar-containing drink, which will essentially give you a fast amount of sugar. What does it do? How does it do it? Essentially, the sugar is immediately processed in the glycolytic machinery. It produces ATP almost instantly. And if you want to sustain this energy, it's got to go into an oxidative machinery. The oxidative machinery gives you, as you well know, order of magnitude more ATP, which will help you really sustain your energy. And so the metabolic family involves a lot of these players. In fact, we have mapped in each of the metabolic disease, in each of the muscle diseases, what goes wrong with metabolism. I want to make one more important point when I talk about metabolism here. The point I want to make here is that when you look at a short distance runner, a sprinter, and a long distance runner, I'm sure you're watching FIFA football matches, and you see that you've got to really gain energy very quickly to run and rapid, running. In fact, I was told that one of the soccer players was running at 25 kilometers per hour in a sustained way. It's really amazing, right? So energy for that is coming from your muscle. And the muscle has to remodel dynamically in order to go from one form to the other. And this is a very important facet of different muscle fibers that are formed in the body. And I don't have much time to tell you about it today, but I can lead you to some interesting articles which will tell you more about muscle remodeling. Now, metabolism is the key towards producing energy. If you recall back my 4-unit diagram, excitation, contraction, coupling, mechanical family, metabolism, and signal transduction, the metabolism, it really plays a key role in providing the energy required for all the other muscle functions. Now, when you look at how does a muscle remodel? So we wanted to analyze what are all the factors that are involved in muscle remodeling, by very painstaking transcriptional measurements and in vitro measurements, in vivo measurements using in situ hybridization methods, we were able to map very precisely what happens once you have excitation, contraction, coupling, and calcium signaling. You get calcium, calmodulin, calcineurin, these are calcium proteins. They in turn activate the class 2 histone deacetylase proteins in the cytosol. These then get transported to the nucleus, and then they activate transcription factors and they bind to chromatin. They modify acetylation patterns. They activate NFAT, and this gives rise to upregulation of slow muscle fibers. This is how you build slow muscle fibers. And in fact, if you want to look at mitochondrial biogenesis, the same pathway activates the PGC1 alpha and then activates the NRF, which activates mitochondrial biogenesis and produces new mitochondria. And so this is a very typical mechanism. And in fact, if you want to look at a developmental muscle or an altering muscle, we'll be able to use these factors in a single cell construct, for example, to look at what stage the cell is going to be in. The metabolic family also, I don't know again how many of you know, but what regulates temperature in your body is the skeletal muscle. The skeletal muscle is absolutely crucial for temperature regulation in human physiology. It does it through this mechanism involving hypothermia and hyperthermia, and these are done through the transcriptional regulation of a very interesting set of factors. And this is what gives rise to activate it through the mTOR signaling network. And this is what changes the body, senses the body temperature and tells the muscle to compensate for either a decreased temperature or increased temperature. And the last thing in signaling and remodeling in the muscle is the muscle under stress can have inflammation signals. In fact, one of the signals that commonly we sense inflammation, for example, when there's an infection, the TNF alpha signaling and interleukin signaling are activated, which gives rise to muscle atrophy and protein breakdown in the muscle. And so these are all the mechanisms by which a muscle remodels dynamically in real time in our human physiology, and we can trace this mechanism. Can we do it in the context of HubMap, where we can really show, for example, vastus lateralis, a muscle in the human? Can we show how this muscle is remodeling? This will be a big challenge going towards the future because this involves really a dynamic perspective in the context of a skeletal muscle in human physiology. Now, let me wind up with a few interesting things. I mean, what happens to atrophy and hypertrophy? As I mentioned to you previously, muscle, when you age, for example, which HubMap does not cover, they don't discuss aging. What happens to muscle when you age? In fact, you know this very well. When people age, the muscles become weak. And in fact, muscles become so weak that it is very difficult. Muscles ache and it's very difficult to do things. Skeletal muscle, exercise, and so forth. So muscle atrophy and hypertrophy are orchestrated through a very interesting set of mechanisms in a cell, in a muscle cell. For example, if you look at insulin growth factor activation, activating through PI3 kinase and AKT, this gives rise to protein synthesis, and this is really the key to maintain the muscle. If when you start aging, many of these factors are downregulated. As a consequence, the muscle starts atrophy, right? And similarly, muscle can also get hyperactivated, hypertrophy of the muscle. And in the case of increased insulin activation, for example, muscle can get hypertrophy, and you have a number of factors like inflammation, for example, which activates the hypertrophic muscle. So this is essentially a schematic way of representing what happens to a muscle through a complex set of events. Okay, so let me now, this was my last slide, but I want to go back to one point here that I want to make. And so I want to go back to revisit this family that I talked to you about. So we have gone from the myogenesis, which is creation of a muscle, from a stem cell, either whether it's an embryonic stem cell from an embryo or from a pluripotent stem cell in vitro, ex vivo. Either way, we walked you through what are the factors that control the growth of the muscle. Once a muscle is formed, then we said, what does the muscle do? The muscle functions through the growth itself is orchestrated through very specific transcriptional machinery and transcription mechanisms. And so now we have a very clear window into each of these stages of growth of a muscle. But once a mature muscle is formed, what does it do? So it senses a signal. Muscle is closely connected to a neuron. So you've got a neuronal signaling, which tells the muscle, nerve activation senses the muscle and sends an electrical signal, which essentially activates the muscle. The muscle, in turn, when it's activated, all it does is calcium. In fact, calcium is the key to the whole process of next steps in a muscle. Calcium binds to troponin, and troponin is the first regulator with calcium, which regulates a whole cascade of pathways, which I walked you through, giving rise to mechanical force transduction through a multiple set, starting from, in fact, when I showed you this picture here, it is going from the outside of the cell to the inside of the cell to again to the outside of the cell. This is the outside of the cell. This is what happens in the calcium signaling. From here on, we are going outwards towards the myosin, actin-myosin subfamily, the Z-disc subfamily, which I showed you. And then we go into transmembranes, which is between the cell and outside, and the ECM, which is the extracellular matrix. So this is what happens in the mechanical signal transduction family. Where do you get the energy for all this activation? And that's where the metabolism comes into picture. The muscle is a very intricate metabolic machine, and it produces in mitochondria the ATP molecules through an intricate set of processes. In fact, we showed that in Lou Gehrig disease, also called amyotrophic lateral sclerosis, ALS, we showed, in fact, that the production of ATP is impaired in the metabolic family. So it gives rise to an alteration, giving rise to hyperthermia, where the ATP is not extruded, so therefore it essentially heats up the system. So the metabolic family is crucial in both maintaining the mechanical force transduction and giving energy for signaling for the chromatin to produce gene expression, to alter gene expression, to change muscle fiber. So metabolism provides the essential energy levels, and the signaling in translation family, I told you, essentially dynamically remodels the muscle fiber. It's involved in temperature sensing, it's involved in inflammation sensing, it's involved in the signals which tell it to produce more muscle, it's involved in the signals which tell it to produce less muscle or atrophied muscle, and it essentially, the commands for the chromatin remodeling are given from the combination of metabolism signaling and the signaling that goes into the nucleus, into the chromatin, to alter the state of the muscle cell. So I hope I provide you a perspective. My whole goal in this was to tell you that while HubMap is providing you with a tissue cell perspective, it does not, we need to add the time angle, the longitudinal angle, which includes the developmental time, which includes the aging time, which includes the dynamic remodeling time within a day, within an hour, within a lifespan of the individual. So if you don't add this, we will not get a full perspective on how a human tissue and cells, crosstalk towards producing function at different levels. So I hope I've convinced you that time is a very important factor in this, in defining HubMap. And we need to have a longitudinal perspective of a tissue. We need to have also the localization perspective of a tissue. The one point I did not have time to tell you, and I didn't have time to walk you through, is that when you look at human physiology, your muscle all over your body, distinct types of muscle. Muscle is one of, in fact, the heaviest, tissue in human physiology. And if you look at the muscle, the different muscle aspects, they are localized in different regions. They are connected to neurons, neuromuscular junction in distinct ways. So context, spatial context is very important. I didn't have time to tell you about the spatial context and regeneration in the spatial context, but this is going to be very crucial for us going towards the future in terms of mapping human physiology. So at this point, I will stop and entertain questions and discussion, and I'll be happy to, walk you through more things. I'll be happy to provide you with references, more perspective in terms of muscle in the context of a human physiology. So thanks for your attention, and please feel free to ask any questions that you have.
Wonderful, Subramaniam. I would like to ask any of our guests to please provide any questions that they might have. Let's give them a moment to collect their thoughts and see what they have to say.
There's probably too much biology for them, not enough computer science. I could have brought in a lot of computer science, but I thought I'll bring in a biological perspective to HubMap.
It is all a part of the Atlas efforts at the end of the day, isn't it?
Yes, it's all building the human atlas. I should tell you, there are NIH efforts now to, there's a very beautiful project called Motor Pack. What they are doing is they're doing exercise, pre and post exercise. They are measuring the skeletal muscle in a large number of human subjects. They are extracting the skeletal muscle, biopsying it, and they're measuring metabolism, looking at transcriptomic measurements, epigenetic measurements on these muscles. So it'll be really fun to have an understanding of what does exercise do to your muscle physiology, which is really a big, big topic.
Professor Shankar, I have one question.
Please go ahead, Supriya.
Yeah. So, in muscle aging and everything, we have food and food habits, or the daily habits of a person really matters a lot. So does it include in the model or network, like how the dietary thing is involved in the muscle aging? Or
It's a really good question, Supriya. So when I talk about metabolism, that metabolism essentially is everything is coming from the nutrients and the food that's intaken. So your metabolic pathway, for example, one thing I did not cover today, and we have done a lot of work on this, is muscle and fat. What's the relationship between muscle and fat? Right, that's a very important thing. In diabetes, for example, or in insulin resistance, you have a crosstalk between muscle and fat. Your skeletal muscle become more adipogenic, right, which becomes more fat-like. So this is where the food and diet becomes very, very important. And this is where your genetic propensity also becomes important for your fat, deposits near the muscle. And so these are really crucial factors for the crosstalk between muscle and fat. Now, just to tell you, NIH also has a very large common fund project called Nutrition for Precision Health, where they are trying 3 different diets for 6,000 humans at a time. So they look at 18,000 human subjects, and they're looking at typical Western diet, a high cholesterol, high fat diet, and a Mediterranean diet. They're feeding them with these things. And so they're then measuring in blood and then hopefully in muscle and adipose, they're measuring changes after, 6 week, or I think it's a 6 week or 9 week diet. They're measuring changes in these. So this is another way of addressing your question, which is what happens to muscle as a consequence of diet. But in principle, healthy diet is a fibrous diet, a healthy diet is always good in terms of getting, activating a muscle or remodeling muscle the right way.
Yeah. Thank you. That answers my question. Thank you so much.
Sure. Do you have any other questions, Devin, online? I don't think you do.
I do not at the moment. I'll just give one more moment to see if we can gather any from our audience.
In most of United States is darkness. It is, in fact, Hawaii is the only place where it is even evening time, late evening time. Otherwise, it's night. In fact, East Coast is very, very, it's daylight, midnight or later. 3 a.m. Ellen has a nice question. Ellen, thank you for the question. Hypothyroidism causes skeletal muscle like to become quite weak, and often one of the first symptoms of hypothyroidism. What is the cause of this? It's a beautiful question. So I've stopped sharing my screen, but one of the things that I mentioned to you is hormones play a very important role in muscle remodeling. Let me see if I can pull up for a quick second, what I shared with you before. Give me 1 second here. If you recall my, let me go to, if you look at this one, look at this slide here. This is what happens in muscle remodeling in human physiology. Look at growth hormones, example, growth factors, hormones, like insulin growth factor, for example. They trigger a whole set of signaling pathways and networks, which gives rise to protein muscle hypertrophy and muscle atrophy. The combination of the two, there's a crosstalk between the two. So you're totally correct. Hypothyroidism, essentially thyroid hormone changes or deficiencies. It causes atrophy. In this case, it causes muscle atrophy. It functions through these transcription factors called MRF, which is, again, talking to a signaling protein called FOXO. And so this is how, in fact, people have done extensive amount of work on hypothyroidic muscle. And this is exactly what they observe as being altered. So activation, in fact, there are pharmacological agonists which activate the FOXO and or which inhibit FOXO and MRF1, giving rise to better muscle maintenance. And I'm sure there are drugs which are publicly available. I don't know these names, but they are. I hope I answered your question, Ellen.
Wonderful. I think that would conclude our segment here. Thank you, Dr. Shankar, for joining us today and sharing your work. Are there any links that you might want to share in the chat?
Yeah, my pleasure. I hope HubMap will extend their endeavor and efforts towards looking at longitudinal and developmental and aging aspects of human tissues, so that we will not just have a perspective of healthy adult, but we want to really have a perspective of a growing child or an aging adult or a deceased adult. And so these are very important perspectives. Thank you for your attention. And if there are any, SENNET, Ellen is very right. SENNET is taking care of aging and studying skeletal muscle. And hopefully in the next 2 decades, we will have a strong body of knowledge that will help us understand physiology from a dynamical perspective. Thanks for your attention. You can have probably a 9-minute break before my very distinguished colleague, Peter Hunter, comes and takes over. Peter is one of the doyens of Human Physiome Project. He's a pioneer in this area. So he'll be really an excellent person to tell you about quantitatively how to look at human physiology. And so that Peter will continue from this point.